This is a production of Cornell University.
Yeah, so I'll introduce our next speakers. Again, this is Cornell researchers and just talking about some of the work that they're doing with breeding. And so we are very fortunate to have Dr. Ginny Moore here with us. Ginny Moore is an assistant professor of plant breeding and genetics at Cornell University. Her lab breeds new crop varieties for organic and sustainable cropping systems, including work on perennial forages, cover crops, alternative crops for the region. Ginny also does a lot of work with looking at hairy vetch. And so if you have questions about hairy vetch and where things are going with variety development there, she's the person to talk with. So I also work with Ginny on some perennial grain crop production, some Kernza work with through her students. And yeah, again, really fortunate to have Dr. Moore here at Cornell University. Also joining the panel today, or as part of this research brief, we have Dr. Solveig Hanson, who is a research support specialist with Dr. Ginny Moore. Solveig joined the lab in January 2023, where she coordinates research and outreach for the nationwide cover crop breeding network. Solveig earned her PhD in plant breeding and genetics from the University of Wisconsin-Madison in 2020, where she conducted genetic, genomic, and participatory research focused on flavor in table beets. In her postdoctoral work at the University of British Columbia, she coordinated a Canada-wide farmer variety trialing network. So a lot of experience working with farmers and doing variety trials and participatory breeding. The other person is Siim Sepp. So Siim is a third-year PhD student in plant breeding and genetics, working with Dr. Mark Sorrells on barley genetics for organic and conventional systems. The goal of this work is to release malting and naked barley varieties adapted for the environmental stresses present in New York State. One of the main goals is to introduce pre-harvest sprouting resistance with elongated dormancy to the breeding program. So very happy to have our researchers here from plant breeding and genetics. So let's give them a round of applause just to help welcome them.
My name is Solveig. I'm a research specialist in Ginny's lab. I work mostly with cover crops, but when I came here, I was glad to join this project, looking at Mississippi and winter lentil production in this area. This project was started by Peter Martin and Kristen, hoping to find ways to diversify both crop rotation and market opportunities for farmers in this area. So we looked at both a winter pea variety trial and a winter lentil seeding rate study. For both trials, we had on-farm or replicated plots on our research stations, so we could do statistics, and then we complemented those with strip trials on Peter's farm to kind of give some context to what we were seeing on the research station. For the winter lentil trial, you could only really source one winter lentil variety, Morton, which is a Turkish red lentil. We looked at different seeding rates. We had 5 rates, which you'll see there. We looked at stand count in the fall and spring, and then we did hand harvest on the research station so we could separate lentils and weeds. First year, we tried lentil monoculture, fall planted, and we found a lot of weeds. And as a result of all those weeds, vine weed and chickweed on replicated plots there, a lot of field daisy and Peter's plots. We saw no significant difference for any trait that we tried to measure because the weed pressure just was too strong. But great learning experience. We figured maybe to get the lentil a fighting chance, we need to add a nurse crop. So for the second year, we did that. We added winter oat, and it worked, right? So well, the pointer's not going to work for me, but you can see that as the seeding rate goes up, so does the plant population. So that works. Using the winter oat as a nurse crop allowed the lentils to establish, and then we were able to answer this question: if we increase plant density, do we also increase lentil yield? And the answer is kind of inconclusive. So you'll see that this table is sorted by lentil yield. And, you know, the highest numerical yield was from the 55 pounds an acre treatment, which was the highest seeding rate. But you'll see that that gave a 63 plants per quadrat, right? Which is about double what we got from 40 pounds an acre seeding rate. But the grain yield didn't increase by that proportion at all. And we didn't see any statistically significant differences between any of these seeding rates. So our best, we don't have great evidence that seeding more than 40 pounds an acre is worth it. We only really have 1 year of data on this, so it's inconclusive. We'll look at the on-farm results. So Peter did get the highest yield at 55 pounds an acre, but the next highest was the 40 pounds an acre. Peter, did you come to a conclusion as to what seeding rate you would use in the future?
Last fall, I planted at 40 pounds.
Okay.
The cost of seed is high enough that extra little bump is much more worthwhile.
Yeah, good stuff. So I looked at lentil yield data collected by the USDA. They collect lentil yield data in like 4 Western and Great Plains states. And those average between 600 and 1,100 pounds per acre. So we're really right in that range, which is pretty cool that we can get those yields here. So biggest conclusions from the lentil trial, use a nurse crop to get them to establish, and we didn't see great evidence that any seeding rate greater than 40 pounds an acre is going to be worth it. For the winter grain peas, we were able to source a great number of varieties. We sourced these from Progene. If I understand your original question, the most commonly planted variety around here is Keystone, correct? There's Jay, who just spoke. You grow the pea. Yeah, what do you grow?
It's an Austrian type. This was focused, I think, entirely on potential food grade types, yellows.
Yeah. Okay, great. So our question was, can we find a grain pea variety that will be better than Keystone for yield? So the first year we saw significant differences for all the traits we measured, except for weed biomass. So that's satisfying. We saw some trends. We saw that I kind of gave these an Olympic medal ranking. So the first group are shorter plants with more stems. They did best for yield and best for protein content. There's a middle ranking. The silver ones are taller plants with fewer stems, also lower protein content, and then some lower performance at the bottom. So, and we got similar results on the farm. So all the varieties in each of these groupings are the same, even though Peter had a slightly smaller subset on the farm. So we were thinking like, hey, maybe we're discovering a real trend. But then came year 2. We our field on the research station was much wetter, much more prone to disease. So the varieties that really did well were the indeterminate vining ones that can kind of grow out of the disease. There was a lot of lodging. Some of the more determinate varieties, the semi-leafless ones, they detached from the soil at the soil level. There was so much soil-borne disease. So that drove these results. The 2 varieties that did really well, Keystone Icicle and Icicle, were those long indeterminate varieties. You can see the strong difference between the plant height and the percent standing, which is, you know, the inverse of lodging. So in different conditions, different plant architecture functions best. Important to realize that harvest was a lot more difficult for these plots because they were so voluminous and viney. We did mechanical harvest on these with great effort with a plot harvester that we borrowed from the Sorrells team. And your harvester did fine. Do you want to talk about what you used?
We have a honeybee flex scraper with an air reel, and I really upset them when I sent them a video of just cruising through the plots, no problems at all.
Yeah, we're like feeding it in with like a push through. It was ridiculous. But Okay, so we, on the research station, we measured yield differently the 2 years. So first year, we had much more controlled plant size, so we did quadrat harvest. Second year, massive plants, we did mechanical harvest. But we could rank which varieties yielded best, or in which order in both years. And you can see by all this crossover that some varieties did much better in the drier years, some varieties did much better in the wetter conditions. So we do find some nice horizontal lines. So Herdwood and Goldenwood were the ones that did well, or slightly above average in both years, in both those drier conditions and the wetter ones. Keystone was more like a relatively stable variety, but it was slightly below average in yield both years. So these are Peter's results, but similar variety ranking over both years. Have you decided whether you're gonna change varieties from Keystone?
I didn't actually plant any this fall because
Okay.
we kind of got and we don't have a market right now.
Gotcha. Okay.
It's something that we probably will look at again. Keystone was good. The issue I've had with Keystone is pea weevil, which we haven't touched on, but it's a major problem.
Yeah, I wonder,
Yeah.
do you think that's different between varieties? Did you?
I suspect that we're going to see pea weevil in any of the yellows or greens because they're more palatable for the pea weevil.
Yeah. That sounds right, that's a whole nother frontier. Also see dramatic differences in yield levels between years. So the second year yields, between 1,000 and 2,000 pounds are also in line with the USDA average yields for the western states. The upper half of them are in line. The first year was a real bust. You had a hypothesis on why that was. I think you were thinking that you grew them with triticale the first year and over the second year?
Yeah, okay, so I think that there might be an allelopathy between the triticale and the winter peas that suppress the peas.
Mhm.
The winter oats are something that we've been working on. We had oat contamination that survived winter in some of our wheat in 2021. We've been developing that line. Oats can have allelopathy, but I am hypothesizing that it does not have an allelopathic effect against the peas, which didn't suppress the peas. The peas were able to do a lot better in the spring when they came out of dormancy.
Yeah, that's interesting because, we grew our research plots with triticale as well, but it was a different variety than we were using, I believe.
Yeah.
So that could be one of those variety crop interactions like we're talking about with sunflowers and rye. Because we've seen that in other allelopathy research that it is very cultivar specific, cultivar and species specific. Yeah,
Another thing to consider, because these are being grown for food production, is that a lot of the lentil and pea markets are looking for, if not certified gluten-free, some assurance that it is gluten-free. And if we're using triticale, that is a glutinous grain. If we're using oats, that is not a glutinous grain. So it may give an easier entry into the market.
That's a great dynamic to consider.
And they're a lot easier to separate because triticale test weight and pea test weight are too similar. Oat test weight and pea test weight are quite different.
Good stuff, thank you. So here are kind of the conclusions from the pea trial. Performance really differed by the amount of moisture and the pea type. So semi-leaflet's more determinate versus normal leafy and less determinate. You have extended results in those handouts if you want to dig into vigor and yield and protein content and things like that. But I will leave it here. I'll take a question or two if there are some while we get Ginny's presentation set up. Anybody else? Yeah.
I was wondering about using winter oats as a nurse crop as opposed to the regular spring oats.
These
They were better. Yes.
I know, but why would you do that? Because spring oats are nice, but they might grow more vigorously when they get die. And leaving everything to be cleaner and easier. I don't know, just a question.
Yeah.
I like to have the oats out there because it aids in harvest. So the spring oat, there's concern of smother when it winter gilts, because especially lentils, growing point remains under the surface. over winter. And if they have a hard time pushing out of that dead mulch, I'd be worried about them getting started. That winter oat also creates just a lot more biomass to pull into the combine so that you don't lose as many lentils to shattering. Because lentils are extremely prone to shattering once they're ripe enough for harvest.
Do they ripen at the same time, or do you have to windrow and look back? I don't know.
This past year was kind of an anomaly on every front, but they did ripen together fairly nicely.
For the lentils, it was definitely ready for me, trying to identify when they're ready, because like the lower part of the plant is still greenish and the upper part is yellowish brown. The pods are brown, but the rest of the plant is not. And if you wait till the whole plant's brown, all the seeds are already on the ground. So yeah, that's something to learn. Yeah.
There is another effect or benefit from the oats, and that's it. It does provide some biology in the soil that suppresses insects.
Mm-hmm.
So in addition to suppressing weeds, it's also suppressing some of the diseases and insect problems.
Excellent, thank you. Yeah, one more?
We used to grow peas and oats, and just for a pretty good separation, it was coarse, it's not like a mill, but with a rotary screen cleaner and had a screen made. I don't know what it was, something by 3/4, so the oats would fall through, the peas would beat through, but the oats would fall out.
That's how we pre-clean them before we bring them into the plant, so that it's less of a load on the plant.
Remember, Ginny, I'll be around later. Do you want to talk? These are lentils.
Okay, it's fine. Being here, and thanks Solveig for talking about our pea and lentil project. I'm going to be transitioning back to talking more about rye, which we've talked about quite a bit this morning already. Before I do that, I just want to mention sort of the broader work that we're doing on cover crop breeding. I won't talk about the other species that we're working on in our lab, but just to say that Solveig and I together lead the cover crop breeding network, which is this national group of plant breeders and farmers and agronomists and a whole bunch of folks all working to develop improved cover crop varieties. And the network started out working on winter annual legumes. So we initially started doing breeding work on vetch, pea, and crimson clover. And then in the last few years, we've added rye and winter canola as additional species in the network. And so the work that I'm presenting today is kind of some initial work that we started doing as we were getting to know rye as a cover crop species and getting to know the genetics and management systems in our lab. And the work was supported by the USDA Organic Research and Extension Initiative, OREI, as well as the Northern New York Ag Development Program. So you'll see that a lot of the work happened in the northern tier of the state. And yeah, so this figure is probably not news to most people in the room, that rye is by far the top planted cover crop in the US. And so that was one of the main motivations for adding rye to our breeding network. It's planted on a lot of acreage and a lot of potential impact if we can improve the species. For a lot of the reasons that Erin mentioned at the beginning of her talk, it's, consistent winter hardy, very productive cover crop. Has a lot of applied in a lot of different ways on different farms. And Erin was also mentioning that the challenge of or the importance of planting in a timely fashion, getting it in, ideally in mid-September, which can be a challenge depending on the rotation that it's planted in. And as a plant breeder, I'm always looking at things with the lens of how do we use genetics as a tool to either improve the performance of cover crops or make it easier for farmers that are not yet using cover crops to incorporate them in the rotation. And so we know, as Erin was talking about, that biomass shows some pretty clear declines with later planting dates. And this has implications for the benefits that you're getting from cover crops, certainly weed management. And this is a figure from an study in Alabama, but we see same trends in other regions as well, including here. The dates are different, obviously. And this second figure is from a meta-analysis in the U.S. Midwest, and we see that at this around 4,500 pounds per acre, that we can see a 75% reduction in wheat biomass. So in this particular set of studies, that was the target. I know Aaron talked about much higher levels of biomass, and that's this meta-analysis I don't think was specific to no-till systems. So certainly for those systems, you may need to achieve higher levels of biomass, and so so certainly depending on the application, you might be looking for different performance of the cover crop. For the purposes of my presentation, I thought about this 4,500 pounds per acre target number, I'd be really interested to hear what other folks are targeting in different systems, and the data that I'm presenting, you're getting the hot off the presses, so this is analysis that I've just started doing the first time I'm presenting it, so I'll be really, I'm excited to get feedback from folks about how you might want to see the data analyzed differently, or what kind of context is important to consider as we're thinking about these numbers. But so my question really is, we started out thinking about whether variety selection can improve performance at later planting. So acknowledging the reality that sometimes the mid-September date isn't realistic in a specific, a given system or a given year. Can we still achieve sufficient levels of biomass to see some benefits, even if we plant a little bit later? And a lot of the rye cover crops that are planted are with variety not stated seed, so we're not using this tool of genetics to target and improve the system. And a lot of the studies that we've seen that have evaluated planting dates are testing just one or very few rye varieties, and so not really accounting for potential genetic variability within the species. So what we wanted to know is there variability among rye varieties that are already on the market for performance when they're planted later, and can some varieties be planted late and still accumulate enough biomass to provide some value for farms? And so we did a planting date by variety experiment over a couple of years. So we planted, here we go. So we planted in 2 years. So fall 2022 was the first year that we planted the experiment. We continued it the next season as well. We planted on 4 dates, so late September, early October, mid-October, and late October to early November. So that first planting date might be in so we targeted around September 25th, which I think is a recommended kind of deadline from NRCS. So all of the later planting dates are in these really marginal times, so not ideal, not recommended generally. And we planted, again, a seeding rate that's lower than what Aaron was describing. So this is also based on NRCS recommendations and not specific to no-till. So it take results with a grain of salt if that's the system that you're targeting. And so you might see higher biomass values at a given date if you're planting at these higher seeding rates that you might need for no-till. But I would expect that the trends may be similar, but at higher final biomass at higher seeding rates. So we planted this experiment at 3 research farms. So at the Freeville Research Farm close to Ithaca, and then at the Extension Learning Farm in Canton, and the Shazy, the Minor Institute in Shazy, New York. We also planted, not a planting date experiment, but just the same varieties on 3 farms as well. I'm not going to present the on-farm data because I really wanted to focus on this planting date question. But just to say that we saw fairly similar variety rankings on the farms as well. And we planted 6 varieties in the first year and then added 8 more in the second year. And yeah, so I'll get into the results. So the first thing, as expected, we saw these planting date effects that in general at the sites we saw declining biomass with later dates. The exception, we had 1 year where in Shazy, where we had some disease issues in a couple of the earlier planting dates, so they had more comparable performance across the dates. There were differences, but they weren't in the order we expected. So that's a caveat for that site. And variety also was clearly important across all of the sites. So variety did matter in terms of the final biomass production. And in most cases, we didn't see an interaction between planting date and variety. This indicated to me that you could plant the same variety. The same varieties were the best regardless of the date you planted them. So you don't necessarily need to switch varieties if you realize that you're going to have to plant late. You would plant the same variety regardless of the timing. The exception was in Freeville. In the second year, we did see some rank changes, although to the groupings remained pretty similar. And okay, so getting into the nitty-gritty of the variety performance at sites. So I'll go over first the sites that didn't have an interaction effect. First, I would say, so in Canton, we had really low biomass production across all of the dates and varieties. So yeah, although the rankings, the variety rankings were quite similar across the different sites, I would call the Canton site a failed establishment across the board. So I'm not going to focus on that data. And I also think that the data in the first year in Shazy is a little bit less interesting because everything did pretty well. So everything surpassed that 4,500 pounds per acre biomass threshold, regardless of planting date and variety. So this data is maybe doesn't really help us answer the question as much either. So it's these remaining sites where we had, we were right at that threshold of the minimum recommended biomass. And so some varieties on some days were performing above the threshold and some were performing below. So the first site I'll talk about, the second year in Shazy. The most interesting planting date was this October 17th date. So this was the 3rd planting date in the experiment. And about half of the varieties still surpassed that 4,500 pounds per acre threshold. And because the threshold, if we change the threshold, we'll see a different number of varieties. But that really, that planting date had the most variability. The data in the table are the average across the dates, but I've highlighted in the boxes the ones that that still passed the threshold on that 3rd planting day. So even in mid-October, they were able to produce a pretty decent amount of biomass. In Freeville, in the first year, at the second planting on October 12th, we only had 2 varieties, Elbin and Andy Gardner, that surpassed that threshold. And then the last site is the one that had the interaction effect. So that's that data is here. And again, so there's some dates. So this first date, all of the varieties surpassed that 4,500 pounds per acre. And then in day 3 and 4, nothing did. But at the second planting date, which was October 4th, again, about half of the varieties passed that threshold. And you can see There are some common varieties that met that threshold across these, three different, marginal planting dates at these three locations. And so ND Gardner and Elbon, in all 3 years, were among the top performers and surpassed that threshold. And Ryan and Renzabuzzi were only planted in the second year, but in those years that they were planted, they also performed quite well. And yeah, so to me, this says that there is, potential for genetics to help push the boundaries and allow, on these marginal dates, suboptimal dates, that you still could, if you plant an appropriate variety, could see better performance than with other varieties. And so then I want to talk briefly about the breeding work that we're doing on this front. So we in addition to evaluating varieties that already exist on the market for this performance at later planting dates, we also want to know if we can actually intentionally breed to improve performance of rye at late plantings. So this is work that's led by my PhD student, Raksha Thapa, and she, as part of this Northern New York funded project, planted a nursery at the Willsboro Research Farm along Lake Champlain, and we planted both at this very more northern location than where we are in Ithaca, and at a later date, in the hopes that we would experience some challenging conditions and really challenge and stress our rye nursery and be able to make some good selections for more winter hardy and productive varieties. We had our first winter nursery there last year, and we've now planted an evaluation of that population, but we also have some populations that we've selected in a controlled environment. So one concern with any of our work selecting for improved winter hardiness or improved germination under cold conditions is that environmental conditions are variable. We can't predict when we're going to have a good selection year, and so we want to develop tools that we can more predictably make good selections. And so we've been evaluating this tool called the thermogradient table to see if that can help us do this work more efficiently and predictably. So this is basically a table that has tubing running underneath, and we have temperature controlled baths that circulate through the table, so we can control the temperature and have quite cold soil temperatures. And we can also have a gradient of different temperatures, so we can evaluate the same material in the same experiment across multiple temperatures. And so we planted a mini nursery in the table, and evaluating germination and vigor at very cold temperatures, so close to freezing. And we even in some cases had like ice formation on the top of the surface of the table, and still had rye plants emerging, which was pretty cool to see. It's definitely a hardy, resilient plant. And so we have a couple of cycles of selection from this breeding approach, and what we're going to be comparing this coming spring, the performance of both the controlled environment selections and the field selections, evaluating it in Willsboro and in the table to see how correlated the selections are between controlled environment and field. So not only can we select things in the controlled environment, but does that actually translate to improved field performance or not? So there'll be data next year, hopefully, at this meeting to tell you the answer to that question. But really excited about this area of research, and looking forward to learning more. And I will leave it there to again thank the folks who have supported this project through collaboration and funding. And I also want to put a plug in. We just this week started distributing a survey focused on rye to guide the rye breeding priorities of this new program. And we're looking for for farmer input on breeding priorities, but also, folks in the seed industry or extension, anybody that's working with people that are producing rye or processing rye or working with rye at various types of businesses and education and things like that. So if you interact with rye, please follow the QR code and let us know what you think. And yeah, we really appreciate your input, and it'll guide the future work that our breeding program is doing. Thanks so much.
You show these guys on three different places.
Mhm.
It's not a thing, just the color on the three of them.
That's a good question. We do have soil data, and I didn't analyze it and include it here. Certainly the soil types are different. I couldn't tell you exactly what the different soil types are at the different locations. I think also like our failed site, I think had some fertility issues. I would guess that's why we had such low biomass production compared to the other sites. But yeah, and the Freeville location is very gravelly. And the Shazy location has pretty nice soil.
If you drive across New York State, 7 hour drive from west to east,
Mhm.
you got about 4 hours from the north part.
Mhm.
So you got constantly different.
Yeah.
You got constantly different variable amount of moisture
Mhm.
that show. And so color is why I think it's big.
Yeah. Yeah, for sure. Yeah, and I was surprised at how much we had overlapping varieties that were the top performers, given the huge variability in the state. And yeah, we know we had very different soil conditions and weather conditions in the different sites, but yeah, it seemed promising to me. I mean, it's only 2 years of data, but promising that there were some varieties that were fairly consistently high performers, even at those late dates.
Hi everybody, my name is Siim. I'm a third-year PhD student working in small grains breeding and genetics lab. Just wanted to mention here, originally born and raised also on an organic farm in Southwest Estonia. So I know what you guys are dealing with. And interestingly, the problems are really similar to what we work with here in New York in terms of breeding. So what I'm mainly going to talk about today is naked barley, but the small grains breeding program established at Cornell is actually coming from 1906, so it's more than 100 years old, which is really fascinating for me, and has a focus on investigating small grains and releasing varieties adapted to the local conditions for a while now. But more than 119 years later, what's happening today at our breeding program is mostly focused on breeding winter wheat, malting barley, organic naked barley, that's what I'm going to talk about today, and also MRI corn and spelt. And meanwhile, we breed those varieties. We're also trying to combine that with the latest research that's available to improve our breeding program as well as we can, with some of those topics that we are focusing, like applied breeding methods, molecular genetics, characterizing different traits that are important, and also determination and malting quality, which has been one of my focuses. Just to kind of give a little bit of background, why we even want to grow barley at all. I guess we've heard a lot about having crops that are out there on the fields in the winter. Gives the farmers good weed suppression, improves the soil health, also enables us to break the disease and pest cycles out there. But I think with barley, there's a little bit more than that. As an example, I think barley is one of the most adaptable varieties that anybody could put out on their fields, has relatively low inputs, and really can survive on a variety of soils and environmental conditions out there. Meanwhile, the end product itself can be used for several different causes, like malting. That has been one of our main interest in our lab, animal feed, and also food production. And to kind of get closer to what I'm going to talk about today, the naked barley, there's two types of barleys that I want to be able to distinguish really clearly. So what we usually use to see out on the fields and use for making malt for our brewing industries is, I would say, like 99% covered barley. It has to hull around the grain. But another opportunity that we're working with a bigger USDA funded grant is naked barley that doesn't have the hull around the grain, and this enables for smaller farmers or local producers to get the more high quality product out of it, as it needs less mechanical processing that could potentially eliminate some of those nutritional elements that we're interested in. So I don't know, has everybody seen naked barley out there? Some of us here. So I'm not sure this laser is going to work, but we could get that going really quick. Here we go. Yeah. So difference between the hulls and half barley is all about the hull, right? And interestingly enough, we have witnessed something in the evolution of barley that has caused a single base pair mutation in the barley, in the gene called the new gene, interestingly enough. And what it has done, it has provided us naked barley that, in a way, that eliminates kind of biological glue between the grain and the hull itself, enabling us to lose the hull of the barley grain during the harvesting already, without the need to go through the polishing or the dehulling process that could lose important parts like the germ and the bran of the grain that have a lot of nutrients. And this would also make it not possible to call the grain product a whole grain anymore if those parts are gone. But why would anybody want to grow naked barley at all? Yeah, I just mentioned easier to process and handle, as there's also no need for this expensive machinery that would cause increased expenses and lose those nutrients in there. And also, I think it's a great potential for a higher market value due to its health benefits, like increased beta-glucan gain, and also those nutrients that we don't lose in the process of dehulling or polishing. And I think it still has a variety of uses that one could find. But what's most fascinating for me is the potential to diversify the crops in the region, and also to address the consumer demand for local organic products, as other barley products or other ways I feel are sometimes a little more complicated to get out to any product to the consumer. So I think this using or growing a naked barley is a good way to simplify the process of product development from the farm to out to the farmer's market or the shelf of the store. So we are part of actually this bigger grant funded by USDA Organic Research and Extension Initiative. It's called Multi-Use Naked Barley for Organic Systems. In there, we have 5 different universities all across the US, and also some regional people that help us to get the knowledge out to the farmers and people in the public. As an example, we have Greenwood Center for Regional Food and Farming here in New York. And this is all true that we had a meetup last year. So what is this project about? It's a multi-year project, and it's covering a lot of goals, actually, to be honest. So like, I've listed them all out here, but to explain in more detail, I don't have that much time, but everybody can read them out there. But overall, what I want to get out from here is to show how to show our like multifaceted approach to doing several things at the same time. We're trying to breed new naked barley varieties for farmers that are adapted to local growing conditions. We want to test varieties that are already out there, and at the same time, we want to increase this knowledge out there that the public would know more about naked barley and its benefits, and also the consumers and farmers and everybody that could come into contact with that. So to conclude, I could kind of squeeze all of those goals into those 3 points that I just mentioned. Identifying the varieties that we could grow now, fine-tuning the barley genetics to generate new variation that's even better, and educating farmers and the wider public about the benefits. So we have actually had those organic naked barley trials going on in Ithaca for a few years. We've been testing winter and spring varieties out there, and here I have just kind of written out a couple of things I've noticed. Performance of winter barley has been really good. No big issues with winter survival. And what's been fascinating for me is that we haven't seen really significant differences in the yield, conventional malting barley varieties or the varieties in the organic environments that are naked. And I gotta mention that we see like a small yield loss, but I think that's mostly coming because the grains that we harvest don't have the hull. So the hull already weighs something, so we can't really think of that as marketable yield. Yeah. We've also grown spring varieties. They'll have had some issues with drought and some drier year, but overall also been performing relatively well in the Finger Lakes region. And we've also had a lot of focus on trying to quantify the resistance for different traits that we have flying around here, like Fusarium head blight, smut, and also trying to keep tabs on the dawn accumulation on the grains, to make sure it's marketable in the food industry. And what else have we noticed about those trials is even the beta-glucan levels in naked barley are high, which is really good for people's digestion. It can complicate using naked barley in malting processes, which is important here in the region. And also that we are not having too many problems with the weed management. I feel that most of our problems are still originating from different bacteria and funguses that live out there and want to share the crop with us. And yeah, due to that, we have kind of developed a breeding strategy with all those other universities together to found this breeding population by crossing different already covered lines and non-covered lines, in terms of just getting those good traits over from those varieties that have bred for, like, as an example, for malting barley or for like high yield for longer. Because interestingly enough, what I need to mention is that those covered and non-covered barleys have been relatively segregated for a long time. As we know that they used to be one before this mutation happened, but I guess now they haven't been crossed to each other too much. So they do have relatively different genetic backgrounds that we can exploit by crossing them with each other and seeing what could help us out. So yeah, the main things we're doing with naked barley in Cornell is growing out different populations within this project. We're growing out diversity population to see is there any exotic or interesting trait that we could map down genetically and exploit that in our breeding program. We're also growing out the breeding population, where we're constantly generating the new variation and selecting on that. And the third fields that we're growing out in Cornell has been those regional trials, where we test our best spring and fall lines all across those locations in the US. So Oregon, Minnesota, Wisconsin, and also Texas, California. And our main focus here has been on the traits that are important for the varieties to grow in New York, which for those lines is winter survival, pre-harvest sprouting resistance, drought resistance for springs, disease resistance, and also weed compatibility that a former graduate student called Kunzi used to do some science on. So here what I want to show is just the overall pipeline that we have been sending those varieties through over the years. And since we are in the second cycle of this project, we already have those lines that we crossed a few years ago. At this point, we already have them running on the trials that we can make better selections. Already for some potential line candidates and test them across the United States. So here I'm just pointing out some of the why it's so important to test those traits here locally. As we see, we might face relatively different conditions as one would face in Minnesota or Davis. So here are just some of the examples what I've witnessed in my fields in terms of winter kill, in terms of adaptation to the different environmental conditions, when it sometimes gets 70 degrees and then it gets cold again. And just that's what we're doing out there, is testing which of those plants really survive, which do not. And here on the picture on the right, you see some of those diseases that are kind of climbing up those leaves in the summer. They're relatively hidden if you look at the crop on the top, but as soon as you look into the canopy, there's a whole life going down there on those leaves that we're still trying to quantify. So my research mostly with naked barley is focused on pre-harvest sprouting resistance. And just to quickly define what is pre-harvest sprouting, pre-harvest sprouting is something that happens with those barley grains on the field. As the moisture increases, they might germinate out on the field, depending on their genetic background, which has been a really problematic thing in South Central New York in July when the rains are hitting us hard. And so one thing is, if it happens on the field, we cannot really measure that every year. So we have developed a method to measure that inside a greenhouse with an artificial misting array, where you just harvest 5 spikes from every genetic line, dry them out 3 days, and put them into the misting chamber for 3 days, and kind of exposing those lines to those humid conditions to get this score of susceptibility to this pre-harvest sprouting. And here is an example. We see it's going to be 0 to 9 score, but here we see how those most resistant lines don't have any sprouting in any of those grains after 3 days of being completely poured on, as when some of those lines have genetic backgrounds that just have almost every grain germinate. And when this happens, doesn't matter if it's naked barley or just malting barley or any beet barley, this is something that no farmer really wants to see in their field, as that would just mean a complete yield loss. But luckily for us, we have been able to really narrow into the genetics of this trait completely. And here I'm just showing some work from our previous grad student, Dan Sweeney, who figured out specific base pairs with a couple of other students that are causing those this variation I just showed on the last picture. So we see that if the barley line has at this specific location, so barley has a relatively big genome, millions of base pair, but if this specific one base pair has A and G there, it does have almost the highest, like as an example, number 7 score. As this NN is like the super non-dormant one. But if the barley line would have no A and G there, and a T there, meaning it's the most super dormant one, we see a relatively massive difference between the sprouting score. And this is something that, so these are actually the 3 genes here. And so these are the different specific base pairs that are in there. And just by measuring this in the lab or sending it out and having somebody else measure this in the lab, we could already just, even without testing those lines out in the field, we could already see which one has the traits that we are trying to select for and which one doesn't. So this is what I'm doing right now. I'm growing the populations out with the naked barley. So once we work that out in the covered barley background, and my approach right now is to take this into the naked barley background and just to prove that this is also working in this background. And we could also have naked barley lines that are suitable for New York and that would not sprout in the middle of July when it would rain too much. And this is where I want to conclude my presentation. If anybody has any more questions about naked barley or where to get the seed, if somebody's interested, come ask me or June Russell right here in the crowd. I think she could also help people out. And yeah, I'm just taking a chance to thank everybody that has been helping me out. Corbett Grass and Carl Kunze, our small grains lab, who's also right here, David Bencher, Priscilla Thompson, and Jason Schiller, that have been big help for us, and also all those collaborators from different universities. And if you have more questions, or you just want to check out our websites, here's the one for that tells more info about the Naked Barley project, and this is just more info about our small grains project at Cornell University. So that's all for me, and thank you.
Any questions for Siim?
You mentioned the variety here, but you also mentioned health benefits for the soil and human health of barley. So you mentioned what goes on.
The health benefit has been, in a bigger picture, related to the increased beta-glucan that is related with the digestive system. And I think it provides those more complicated carbohydrates that are, in terms of writing, like, I would say, that improve the microbiome in the gut, and yeah, just make the system in the gut, I would say, more stable. Yeah, I'm not a medical researcher, so can't quote me on that, but yeah, that's as far as I know.
Soluble fiber.
Yeah, soluble fiber, there you go.
This has been a production of Cornell University, on the web at cornell.edu. This has been a production of Cornell University, on the web at cornell.edu.